So hello everyone, welcome to the Network Physiology webinar. My name is Alireza Mani and I'm one of the organizers of a series of webinars on the emerging field of network physiology. This webinar is the first session of this series. Each session will feature two talks, with each talk lasting approximately 25 to 30 minutes. After the second talk, we will have a question and answer session, and we all hope that you enjoy these sessions and you found them very useful. I would like to thank all the speakers who accepted our invitation, and also the Physiological Society for making these events possible. My colleague, Dr. Tope Oyelade, is the other organizer of the event, and I would like Tope to say a few words. Tope, would you like to say a few words?
Yeah, thank you very much, Ali, for that brief introduction. Welcome everyone again. I will probably, before we start inviting our speakers to talk, I'll go just through some of the housekeeping instructions. Please be aware that this webinar is being recorded for the purpose of publishing it in future. So we kindly ask you all to keep your microphones and video cameras off for the duration of this meeting. Today's meeting will go over the general overview of the field of network physiology, both in terms of history and in terms of clinical application. We know definitely that some of you are going to have a lot of very interesting questions. We're really looking forward to it. But please submit any questions you have through the chat. Questions can be typed at any time during the presentation. All questions would then be directed to the speakers and chairs during the Q&A question and answer session that's going to come at the end of the talk. So I'll let Ali to go over the introduction of our first speaker. Thank you.
Sure, of course. Thank you, Tope. Thank you very much. So it is my real pleasure to introduce Professor Plamen Ivanov to you. Professor Plamen Ivanov is the director of the Keck Laboratory for Network Physiology at Boston University. He is the originator and founder of the multidisciplinary field of network physiology and has pioneered the study of the dynamic network interactions among physiological and organ systems. He has introduced innovative approaches to analyzing and modeling physiological systems by adapting and developing concepts from modern statistical physics, nonlinear dynamics, and network theory. Professor Ivanov has a background and a master's degree in condensed matter physics and holds a PhD in cellular biophysics from Boston University. He is one of the 9 founding members of PhysioNet, an international research resource which has been a cornerstone for open access physiological data, computational tools, and algorithms. From all of us who use PhysioNet, we are very grateful. It's such a interesting and good platform. Professor Ivanov has been a leading figure in interdisciplinary research. His contributions to the field has been recognized with several prestigious awards, including the Sustained Research Excellence Award at Harvard Medical School, the Georgy Najakov Medal from the Bulgarian Academy of Sciences, and the Pythagoras Award for high achievements in interdisciplinary research. He is also an elected fellow of the American Physical Society and the field chief editor of the journal Frontiers in Network Physiology. His discoveries have been featured in media, including Scientific American, New Scientist, Physics World, Washington Post, the Boston Globe, and so on and so forth. So we are very honored that Professor Ivanov has accepted our invitation to speak on this new field of network physiology and the human physiolome: discoveries, impact, and perspective. Prof. Ivanov, thank you very much.
Thank you very much, Dr. Mani, and thank you and Dr. Oyelade for organizing this webinar. And of course, many thanks to the support and help from the Physiology Society. And thank you all for joining this webinar. Hopefully, it will bring some useful information in one way or another. So let me perhaps start sharing. Talk. Is this okay now?
Yes, thank you. That's perfect.
Thank you.
Thank you.
Yes.
Oh, everybody can see that. Okay. The human organism comprises various organ systems. And the brain, the heart, lungs, kidneys, skeletal muscles, eyes, all these systems are characterized by very complex structural and dynamical organization with their own specific mechanisms of regulation, different scales of activity, and not surprisingly, research and medical specialists in the last decades have primarily traditionally focused on understanding single organ systems. And indeed, these systems are complex and very complex type of structural network underlie the dynamics and function of such system. Here is a couple of examples. If we consider the heart, this is a network of the vasculature. And this network is, of course, embedded in the network of the myocardial tissue with a particular anisotropic network properties. Within the myocardial tissue, we also have the network of Purkinje fibers, this green network here on the right, and practically none of those networks have been fully understood or actually detailedly quantified in order to understand their functions and the role in the contraction of the heart. One can go to the kidneys, and they at different time scales of observation, they have a complex cortex that branches at smaller and smaller scale. Finally, to have on the tip of this branching structure, the glomeruli, where actually blood is being purified. There are millions of those. And how do they synchronize and coordinate their function as a network for the whole kidney to function? This is still unknown. If we consider the lungs, this is a superposition of two networks, the network of the airways and the networks of the arterial and vasculature network, again superposed spatially with a very complex branching process that have been studied in the last years and still are very object of investigations. And this network goes down to the bronchial trees up to the level of the alveolus vascular network. So if you go to the brain, of course, we have a complex structure of various type of networks, a network of the vasculature, a neuronal network. This is, let's say, tensor imaging of the flow, representing the network of flow in the brain. Of course, we have networks superposed, multiplex of networks of neuronal cells of different type of populations, glial cells, astrocytes, and still we do not understand how, what is the structure and how this structure leads to emerging functions at the cortex level and functions and behaviors at the organism level. So in addition to the complexity of individual organ systems, the human organism is in fact an integrated network of these networks. It's a multiplex of networks where these organ systems continuously interact with various feedbacks on different multitude of timescales. For example, stop breathing for a while and your heart rate will increase racing. So these interactions are essential, absolutely essential to maintain health and to generate distinct physiological states. Alterations or breakdown in some of those interactions can lead, of course, to dysfunction of individual systems or collapse of the entire organism. If one of those systems breaks down, for example, cardiac arrest, then this could lead to a cascade of failure to multiple systems and then to finally failure of the entire organism. But it's also possible that even when individual systems are intact, but breaking the underlying both dynamical and biochemical interactions between organ systems can lead to breakdown of the entire organism. So healthy, intact organs are not sufficient for emerging health. And so despite the broad clinical implications for conditions, multiple conditions, including cardiac arrest, coma, multiple organ failure, sepsis, and so on, we do not understand the basic physiological functions and the interaction of organ systems as a network and how physiological states and behaviors at the organism level emerge out of these network interactions. Of course, in the last decades, systems biology and integrative physiology have taught us a lot about the underlying signaling and feedback processes that connect the subcellular, cellular, and tissue level. But in addition to that, we have, in addition to this vertical integration from the subcellular, cellular, and tissue level, there is a horizontal integration among organ systems that is absolutely essential for emerging of physiological state and function. So one fundamental question is to understand indeed how physiological states and functions at the organism level can emerge as a result of complex interactions across scales among diverse physiological and organ systems. And even more, another fundamental question would be, would there be a blueprint reference network that specifically uniquely underlies every physiological state? And then we transition from one state to another. How does this network potentially reorganize how these cross communications between organ systems change to adapt and to facilitate a new function at a new physiological state? So as a result, a new field has emerged in the last decade called network physiology, where the focus has shifted from single organ systems to the network of organ interactions. And so after this introduction, I would, to the rest of the talk, I will give you specific examples to test some of those hypotheses. And again, the hypothesis is that there is a particular laws of cross communication among organ systems that we still do not understand. It's another aspect of physiology that we need to learn in order to understand how organ systems synchronize their dynamics, in order to coordinate functions, in order to have an emergent behaviors at the organism level states. And then the question is with disease, how these behaviors break down. So this is the focus of this new multidisciplinary field. And of course, to understand and to address such a question, one would need to have a continuous high frequency data output from multiple systems in a synchronized way. And here is a sort of Leonardo da Vinci inspired diagram where different nodes represent different systems. This is the heart, represented by the output signal of the heart rate, respiration, chin muscle tone, leg muscle tone, even eye movement, and different cortical rhythms from delta, theta, alpha, beta, which have different role in physiological function. Sleep laboratories, and I used to work and affiliated with sleep laboratories at Brigham and Women in the past at Harvard Medical School. The sleep laboratories provide an environment, both research and clinical, in order where we have continuous streams of synchronized data coming from the brain, muscle tone, respiration, heart dynamics, oxygen concentration, and multiple other parameters. And so this presentation will demonstrate how could we approach this question of cross-communication between systems and what can we learn out of such extracting information about network interactions. And so we will consider, for example, different sleep stages. These are well studied and defined physiological states where we know something about autonomic regulation. We know something about the behavior of the individual organ systems across wake, REM, light, sleep, deep sleep. And still the question is, okay, how do systems cross-communicate? If we look and have a very experimentalist approach, and if we look in high-frequency recordings of data coming from the brain, for example, these are sigma rhythms, and heart rate data and respiratory breathing data, and for example, eye movement data. One can see that these are very complex dynamical signals, but there is certain synchronous activity. When there is a burst in the one system, is followed with a burst in the other, burst in the other, and burst in the other. Across multiple systems, there is a synchronization in bursting activity on multiple locations for over long periods of time along the horizontal axis. So this indicates and triggers the hypothesis that there is a certain crosstalk between systems. There must be certain causality of interaction, and perhaps we could measure their crosstalk and their coupling and network interactions through a measure of the time delay with which they cross-communicate. If they are coupled, they will have a fixed time delay of response. If one system activates, the other one, if it's coupled with the first one, will have a response with a certain short time delay. And if there is an established link of interaction between physiological interaction between these systems, this time delay would be very stable. And there will be a period of stable time delay, what is shown here on this diagram. If we quantify these time delays, we can see that there are periods of time when these time delays fluctuate, and then for some reason, they stabilize. So a link is formed between two systems. And if we look at random pairs of systems, for example, chin muscle activation with alpha rhythms in the cortex, or heart rate dynamics with eye movement, we could see that there is a time delay in the bursting activity of these systems that fluctuates in an idiosyncratic way for a certain period of time, and then at some moment stabilizes. And for the other pair, it is the same. And so when we consider this short window of about 4 minutes, we can address this question about through the simple concept of time delay as a representation of stable time delay as a representation of interaction between organ systems. How does this network evolve in time? And so this gray scale corresponds, the gray area corresponds to 30 seconds network representation where the subject is in deep sleep. And then here there is a transition to light sleep. And we see that in a very brief period of time, a few seconds, there is a reorganization of the interaction between these nodes, which represent different organ systems, and the network becomes far more connected, more dense, and new links appear, the red one and the yellow one representing a connection between this pair and this pair of physiological systems. And of course, this is just 4 minutes, but what about 4 hours of continuous data recording of a particular subject? And one starts to see a certain consistency in behavior, where the red curve here represents the different physiological states, deep sleep on the bottom, light sleep here, intermediate, light sleep, REM higher up, and wake. So one can see that deep sleeps are always associated with low connectivity represented by this black curve. Low connectivity, deep sleep. The next stage, again, we have a deep sleep, low connectivity. Deep sleep, low connectivity. Again, deep sleep, low connectivity. And if you take light sleep, then we have higher connectivity. Again, another segment of light sleep, higher connectivity. Another segment of light sleep, higher connectivity. So this led us to the hypothesis that perhaps different physiological states are characterized by different network topology representing the dynamic interaction between diverse organ systems. And what was found that during wake and light sleep, we have higher connectivity and lower connectivity during REM and deep sleep. And these diagrams represent average of 35 healthy subjects. And so here is a bar chart that represents the structural change in the network with high number of links, high connectivity for wake and light sleep, and lower for REM and deep sleep, where the red bars represent individual subject and the black bars represent the entire group. A very consistent pattern of change in response to transition across basic physiologic states. And this is pattern not only in the connectivity of the network, but also in the average link strength, which also shows the same stratification. So one could ask a question, well, by the way, this is completely unexpected in the sense that usually when we consider individual physiological systems, light and deep sleep are states where individual physiological system, whether respiratory, cardiac, or brain, exhibit similar properties. And that's why very often they're put together as a state called non-REM, while REM and wake, in some sense, exhibit also certain similarities. From point of view of network interactions, we find that both network connectivity and the strength of the interactions are more expressed for wake and light sleep, and less for REM and deep sleep. So this is a different type of stratification that we observe, which is not the typical classification of sleep stages that we have based on studies, both research and clinical, on understanding individual different individual dynamics of systems across sleep stages. So now, how does this transition from one connectivity and from one link strand to another occurs in the context of transitions across sleep stages? One can assume this could be a idiosyncratic random loss of certain networks or a random decline of the strength of the interactions, or it could be also potentially hierarchically structured. And the answer is the second. If one takes a single node, for example, the heart rate, how the heart rate is connected with other systems during wake, REM, light sleep, and deep sleep, one finds exactly the same stratification pattern, both for individual subjects, showing universe, as well as for the entire group, showing certain physiological universality and indication of underlying physiological law that regulates this dynamical network. If we take the chin muscle activity, for example, how it is synchronized with other systems, both cortical rhythms and other organ systems, again, the same pattern, stronger interactions and higher connectivity for wake and light sleep compared to REM and deep sleep. So therefore, there is a certain hierarchical reorganization that happens in a very structured way in physiological organ networks at the moment of transition from one physiological state to another. So these are, of course, very coarse-grained type of network where each node is, each organ system is represented as a node. But of course, there is a very high complexity behind each node. So if we take, for example, just the brain, how the brain responds to changes in connectivity and strength of interaction, respond to transitions across physiological states. So we have here in sleep medicine, they usually take 6 channels, 2 frontal, FP1 and FP2, 2 central on the cortex, C3 and C4, and 2 occipital. And each location is represented by a small rosette of different cortical rhythms, delta, theta, alpha, sigma, gamma. They have been extensively studied, especially not only in the context of sleep, but in context of whole line of physiological states, in the context of memory and many other functions. And so one can see that during wake, again, we have these strong links with dark blue and magenta and high connectivity, and also during light sleep, and much lower connectivity for REM and deep sleep. And even more so, we see certain modules which are overexpressed with strong interaction, frontal, central, but not so much occipital, or central occipital in the context of deep sleep. So if we quantify this network topology and connectivity from point of view of number of links and average link strength, again, within brain functional networks, we find the same law of regulation, the same sleep stage stratification pattern with higher connectivity and higher strength of interaction for wake and light sleep and lower for REM and deep sleep. So if we extend beyond once the networks, dynamical networks underlying one single system, one can ask how 2 systems interact. And there is this radar chart representation of how cortical rhythms in 6 locations, left hemisphere, right hemisphere of the cortex, coordinate and synchronize activity with chin muscle activation. And so all these links here, for example, these connections are added as vectors on this radar chart to show the total strength of interaction of this area of the cortex, of all cortical rhythms with chin muscle tone, and correspondingly the same with another area of activity. So these vectors show that there is a frontal interaction is stronger because they reach to the edge of the hexagon, and occipital interactions are weaker. Now, if the size of this hexagon would tell you how strong are the interactions, and if we go along with such a relatively simple representation of dynamical interactions of cortical rhythms across locations with chin muscle tone, we see surprisingly the same law. Very high, large hexagons, strong interactions for wake and light sleep, and weaker interactions for REM and deep sleep. And even more, one can ask, what is the hierarchical organization of these links? Are there any particular profiles of links that stay behind? But before that, this is just a slide that shows in the same physiological state how the brain interacts with different peripheral systems, the chin muscle tone, the heart, respiration, eye movement. And so these interactions, even in the same physiological state for the same subject, exhibit very different patterns and different underlying dynamical networks as represented by this schematic diagram of radar charts. So one can extend this, of course, for multiple peripheral organ systems and see how this complex network connectivity between cortical rhythms at different locations with the peripheral organ systems, how it changed with physiological state. Here, for example, the heart is indicated by red because this shows that the main channel of cross communication between the heart and cortical areas is through high frequency cortical rhythms, gamma 1 and gamma 2, while, for example, the eye node is represented by blue, which indicates the main mediator of interaction between cortical activity and eye movement and control of eye movement are low frequency cortical rhythms, delta and theta. And so this network again follows the same, surprisingly, the same rule that higher connectivity for wake and light sleep, lower for REM and deep sleep. One can extend and look only at the horizontal interaction without these vertical brain organ interactions, but also the interactions among organ systems. Here are 5 systems, just as an example, with about 10 links, and one can see how the topology of this small network also changes in a similar way with stronger interactions for wake and light sleep and weaker for REM and deep sleep, and actually much sparser topology at REM and deep sleep. So it turns out that this the whole body is an orchestrated network of networks where different subnetworks adjust to dynamical change across physiological states. So this movie demonstrates a time segment of about 4 hours of data where a subject sleeps, and these are the segments of different physiological states from wake in yellow, REM in red, light sleep in blue, and deep sleep in black. Here are the major subnetworks of the organism: brain functional, brain peripheral organs, and organ subnetworks. And we will see how their topology, and also here these bars show the strength of interaction change across transitions with physiological states. And we observe a very orchestrated, synchronized change both in network topology and interaction, as well as the strength of the links. And when we have wake and here right now you have deep sleep, low connectivity and weak interactions. And when you have REM, for example, again, low connectivity, weak interactions. Now, when we go to light sleep, for example, here, now the connectivity as well as the interactions increase. So there is a very orchestrated behavior among physiological systems that underlie physiological states and the transitions across states. So what is the physiological use of this? If such observations have physiological importance, perhaps we could understand something about aging. So here is several age groups, 5 age groups, 20 to 34-year-old, 35 to 50, 50 to 60, and above 80, where each network, these are brain functional networks, which I showed before, stratified from wake to REM sleep, light and deep sleep, with the same stratification in connectivity, up-down pattern, and also in the average link strength. That's what we have seen on the previous slides. But with increasing age, we see a gradual decline, even though the stratification pattern, this law of regulation with transitions across states that regulates the state of the network, is preserved. So we have a similar stratification also with increasing age, the strength of the interaction, and also the topology of the network changes. But this information is very essential because it tells us that aging doesn't happen in a certain idiosyncratic random way, but certain modules are preserved and certain basic laws of regulation are still preserved in healthy aging. But of course, the general connectivity declines. Still, we have the same modular structure here. For example, frontal to frontal interaction is dominant, as well as here in very elderly subjects. Frontal to occipital here is dominant, and we see it also for the young subjects. So one can extend this to brain organ interactions, and this is a similar radar chart that represents the brain-heart interaction with the same stratification, large network connectivity and strong interaction for wake and light sleep, and weaker, smaller hexagon for REM and deep sleep. And this pattern, up, down, is preserved also with age going down across different age groups. This is 65 to 80, and this is above 80 years old, where we have the same stratification, even though the interaction is reduced, since we see that the size of this hexagon becomes gradually smaller and smaller with aging. Now one can start obtaining dozens and even hundreds of maps representing brain organ interactions. This is for the brain-eye interactions, also changing how they change with age across states. Then brain respiratory system, brain muscle activity, for example, of the leg, and so on. So, of course, these are coarse-grained representations. And for example, here we have the brain muscle tone of the leg with different cortical rhythms across 6 locations of the left and the right hemisphere. But behind this diagram, this radar chart, actually there is a very, very complex network. So now we have interactions between cortical rhythms across locations. These are the cortical rhythms represented by the rosette across locations with the activity of the EMG, electromyographic activity of the leg. And so this radar chart, actually, behind this radar chart, there is a very complex network that looks like this, where we have the rhythms embedded in leg activation derived from EMG. And so these are the rhythms in different areas of the brain. And now again, the same complex network exhibits similar pattern with higher connectivity, thicker links, and more links during wake and light sleep, and much reduced connectivity and strength of link for REM and deep sleep. So across levels, from a very coarse-grained graphs that I showed at the very beginning, where each system was represented by a single node, to the second level, where each system is a network, to the third level, where we have network of networks, what we have here, we see across systems and across scales, we see the same response, orchestrated response, the same physiological law that dictates a higher connectivity and stronger interactions for wake and light sleep, and weaker connectivity and weaker interactions for REM and deep sleep. So these type of networks can be further dissected to understand what is the hierarchical organization of these links. And one can find that these are the links representing cortical rhythms across different locations, FP1, C3, and T1, during wake, during REM, during light sleep, and during deep sleep. And this profile of interaction of cortical rhythm with the chin muscle tone, for example, in this particular case, has a particular profile that shows much stronger interaction that is mediated through high-frequency cortical rhythms. And this profile changes depending on the physiological state. So now we have a wealth of information, a new biomarkers derived from network interactions that can define a state and potentially can define disease. So same, we can consider organ-organ interactions. So this is what I showed before for about average representation of a young group of subjects, 50 subjects, 20 to 35-year-old. And here is the average representation of similar group of elderly subjects older than 80-year-old across wake, REM, light, and deep sleep. And you see how the connectivity changes. And this connectivity is higher, of course, and in the younger group compared to the old. And so this is quantified here. So the profile of all 10 links that we have in this small network is plotted here. And this profile is gradually preserved, but also gradually modulated and declines with age. So you can see the profile, which is a new information, and how this profile declines with aging. So this decline is very structured, again, not idiosyncratic, and now we understand something about the process of healthy aging. So what about major neurodegenerative disorders? Can network physiology help understand, for example, the physiology of Parkinson's disease? And one can go back to the same functional brain interaction representation across cortical areas and cortical rhythms. This pattern that we show with higher connectivity for wake and light sleep compared to REM and deep sleep, we see a similar pattern in Parkinson, but with much expressed and connectivity and strength of interaction. So it's an underlying very strong synchronization behind. So this leads to a change in the sleep stage stratification pattern from healthy to Parkinson. Of course, there is a very high increase in the connectivity and a significant increase in the link strength. So this is what is happening in the brain. What is happening in the brain-heart network interactions, this is what we saw before for a healthy group with higher, stronger interactions for wake and light sleep and weaker for deep sleep and REM. And now we see how these interactions, cortical, cardio interactions, decline in Parkinson. The diagonal of this radar chart significantly declines. Same thing is observed for, let's say, brain-leg muscle control. So with Parkinson, muscle control is reduced, and these interactions can be quantified, and we see the degree of decline. So the profiles that we saw before, indicating the cortical cardiac interactions or the cortical muscular interaction with specific profile characteristics for wake, REM, light, and deep sleep in healthy behavior. Now we see this significant decline that happens in Parkinson in both examples, for example, cortical cardiac and cortical muscular interactions, very significant decline with 30–40%, and loss of this typical stratification pattern that we see in healthy regulation. The cortical muscular networks I showed before across these physiological states in the healthy group are now very dramatically modulated. You have a drop in the connectivity and also in the link strength. One can see also in horizontal interaction among organ systems how these interactions decline. So this is the pattern of interactions in healthy group across states, and a very significant decline in this pattern and modulation of this pattern with Parkinson. This can be extended also to ideas to exercise, something new subfield that is developing now called network physiology of exercise. If we study, we still do not know in the context of exercise physiology how different muscles, we have about 900 skeletal muscles, how this gigantic network, and each muscle is also a network of different muscle fibers and muscle bundles, thousands of them. So how do they internally within the muscle synchronize activity? They're driven by different motor neurons with different frequencies and dynamics. And so here we have a network representing the frequencies of different motor neurons and different muscle fibers during a task of a push-up, where the arm activity, the left and right arm and the left and right chest muscle are integrated into a network. And we can see how this network gradually changes during the duration of the exercise from the beginning of the end, and therefore a new network-derived markers of fatigue, accumulated fatigue, can be quantified and estimated. One can even extend this for protocols where you ask the same subjects to perform exercise, then they rest, same protocol of exercise, they rest again. And even though they rest, one can identify certain accumulation of fatigue. So these networks dramatically changes. One can see the strength of the interactions goes down to more pastel colors instead of the dark red. So the whole vision is, it took in medicine and physiology the last century, since the time of Leonardo da Vinci and before, to identify the human body as a structure of various organs, the locations, and to develop the human anatomy, which is a static representation of organ locations and structure. And the idea is to have a dynamical atlas of most probably now we think it will be millions of maps across scales, from the system level, tissue levels, to the cellular and even metabolic and proteomic level, where we need to understand how all these systems across scales dynamically interact and integrate functions to produce physiological states. So, we need this data to, therefore, to understand, we need data, continuous recordings from multiple systems. We are witnessing the last 2-3 decades that by just scoring the human genome and knowing the content, and these are about 3.4 billion base pairs in the human genome, there was an explosion of studies and understanding of the human organism and various species from point of view of systems biology. So, and this is a gigantic database of information. It's still daunting task, which we've not fully understand. As people say, about 4 billion base pairs, this is about library of 1,000 volumes with 1,000 pages each covered by letters of the nucleotides. So now, if you take in a single day output data of the human body coming from the brain, EEG or fMRI or blood pressure or ECG or core body temperature across locations, muscle functions, hormonal function, kidney function, and so on, if you take just 100 parameters and record them with 100 hertz frequency over one day, you have a magnitude more of data points than in the human genome. Over lifetime, these are astronomical numbers. So can we utilize this information to improve understandings of basic physiology and clinical medicine practice? We need to build something we call the human physiolome. We started a few years ago, the human physiolome project, which would contain thousands of maps representing various conditions, age groups, different physiological states, and hundreds and thousands of diseases. And not only to have the graphic presentation, but also to understand which are the critical zones, the parameters that allow within which we maintain health and beyond which we start to have manifestation of disease. Numerous implications from novel biomarkers to integrating various devices in intensive care units with more comprehensive estimation of drugs, which are usually targeted for particular cellular populations or tissues or organs, but are not tested on their effects on interactions among systems. And of course, new type of education we need, where physiologists and also clinicians pay more attention on understanding how systems interact. For example, in the intensive care unit, instead of tracing the output of individual systems by separate devices, we need a platform of integrated devices that can synchronously record data and tell us something about causality dynamics of these networks. Not to mention implications for robotics and cyborgs and human-machine interactions. So this is a growing multidisciplinary field, and I thank you for your attention. We have started in the last years a new journal which is devoted to understanding the physiology, the network physiology of the human body, and now growing. And we have regular events we organize in Como, in a beautiful place like Lake Como. So I welcome you all. This summer we have an event coming in July. So thank you very much for your attention, and thank you to the organizers again.
Thank you so much, Professor Plamen Ivanov. I think the best way to put it is you're doing amazing work, really. It's incredible. And if there's a way to clap, absolutely. But thank you for sharing your length and breadth of knowledge with us. And it's exciting in the field of network physiology. There's a lot to look forward to, and thank you again. It's incredible. Absolutely. Thank you.
Thank you for inviting me.
Thank you. So It is with pleasure. Of course, we have to move on. It is with pleasure to introduce Dr. Alireza Mani, who is one of the co-organizer of this event. Dr. Mani is an associate professor at the UCL Division of Medicine and is deeply engaged in interdisciplinary project as both a researcher and educator. Fantastic educator, so I must add. His primary research interest lies in understanding the complexity of physiological control in critical illness, successfully integrating computational network analysis with clinical medicine. So Dr. Mani studied medicine at the Tehran University and moved to UCL through a Wellcome Trust Fellowship. He holds a PhD in clinical pharmacology from UCL and leads an interdisciplinary lab where undergrads and postgrad students from diverse scientific backgrounds collaborated on innovative projects. Through his academic career, he's worked to bridge the gap between clinicians and scientists, fostering a common language to drive new advancement and innovation in medical research. His previous appointments include assistant professor at Tabiat Mudaris University and visiting professor at the University of Padua. Dr. Mani is one of the organizers of this webinar, like I said earlier, and we'll be speaking today about the potential application of data-driven physiological network mapping in clinical practice. Thank you very much, Dr. Mani, and you may take the floor. Thank you.
Thank you very much. Thank you, Tope, for a very kind introduction. And now I want to take an opportunity to thank Professor Ivanov. Thank you for your inspiring lecture. It was so fascinating. Thank you. So I want to talk about application of clinical physiology and network mapping in clinical practice. Of course, it is about potential application, but also it is important for us to know that, okay, so based on the common and routine physiological signals and data that we have got, is it possible to use network physiology and all of those great ideas to help patients? I think that's the main idea of my talk. I hope my slides can be visible easily. So, and in particularly, I want to talk about complex diseases such as sepsis. Complex diseases are diseases that are complex interaction between multiple intrinsic and extrinsic factors. We have so many of these interesting diseases, interesting to study. Of course, many patients suffer from it, unfortunately. Sepsis is one of them because sepsis is associated with multi-organ failure. It means that different organ systems just fail in terms of their function, and we don't know exactly why. And then, of course, it causes mortality. It is a life-threatening condition. There is a very interesting and a very sad statistics that 20% of global death is because of sepsis. That's huge, and we never think about that. Think about amount of suffering. So then the idea is we think we know a lot about sepsis because if you look at literature, there are thousands of papers every year, but those data are basic data that are basically, there are lots of local information. So for instance, if we think about sepsis, there are lots of studies on specific, how mitochondria loses its function, or what about the mechanism of cardiac dysfunction, or what about the mechanism of respiratory failure, or so we use many cell cultures or many experimental models, etc., to study that. Use basically reductionistic approach, which has been successful in the past, but maybe for complex systems such as our physiology, maybe it's not enough. So when we think about a reductionistic approach, we are trying to actually break down the system into its pieces, study them very, very detailed. But our local knowledge might not be really good enough to see how everything works together, because actually, of course, we are talking about the whole system. And in sepsis, for instance, almost all clinical trials have failed. In the past 20, 30 years, there is no advancement in actual clinical management of patients in terms of new therapies, although we have lots of knowledge, but these are not enough. Why these are not enough? Can we use network physiology to help and to understand it better or not? I want to give you one maybe analogy. So analogy is that imagine this is the map of London Underground, and my office is around here, and basically this is where UCL University College London is in the Bloomsbury area. I hope you all come and visit our universities someday. Basically, if I have lots of knowledge, local knowledge about the area, all of the shops, all of the little roads, and all of the blue plugs here, it is not going to help me I can get to my collaborators at Imperial College who live in the west of London, or my other collaborators at King's College who live in South London. So if I want to really access London, I need to know the network, the map of the underground, map of the transport of the of London, and just knowing the local knowledge is not going to help me. So it is very similar comparison with understanding the physiology, as Professor Ivanov also mentioned it very beautifully. So there is something very interesting that I want to tell you. Basically, if I want to use routine clinical data for physiological network mapping, so can I use routine data? For instance, of course, we are aware that we have lots of interesting fluctuation in heart rate variations. We call it heart rate variability. And of course, why we have these heart rate variations, it is, of course, maybe reflecting the interaction between different organs. Of course, respiration affects the heart rate, and also baroreflex too. We have all of those interesting intrinsic and extrinsic mechanisms that fluctuate. But going back to previous, of course, studies, going back to 2-3 decades ago, there are interesting studies to show that if you want to ideally map a network of interaction in the physiological system, okay, so maybe it's a bit difficult. Maybe it was difficult 3 decades ago. But okay, so what if you look at the regularity of these time series? These irregularities and these fluctuations might reflect the network. And there are studies based on simulation from 3 decades ago that whenever we have uncoupling of the components of network, it comes with increased regularity. And it is interesting. And it has been studied a lot, actually, in the past 2-3 decades. Basically, of course, there are the wealth of studies on heart rate variability that, of course, normal heart rate variability, we have lots of fluctuations. And then when we have a disease such as, I don't know, sepsis, liver failure, and or even the normal process of aging, then we see more regular cardiac rhythm. It is more regular. It is, when we talk about more regularity, basically, of course, it means that, of course, we can measure it. There are lots of ways to measure it as well. But this decomplexification is interesting. Does it reflect the impaired network connectivity or not? This is something interesting. So we should be able to quantify these variations using a method. And there are loads of different methods established, from simple measurement of standard deviation, which has been used in clinical practice for years and years, to more advanced, such as entropy analysis, which I will talk about a little bit later. But we should not really restrict ourselves to heart rate variations. So basically, heart rate variability has been studied a lot, but there are so many other interesting physiological rhythms, physiological signals that, of course, fluctuate and give us lots of information about the integrity of physiological system. For instance, skin body temperature. We don't use it that much in clinical practice, but it is very interesting because it shows the balance between heat loss and heat gain. It shows a lot about, okay, integrity of the vasculature, etc. So, and there are recent studies to show that assessing this fluctuation can give us lots of information about health and disease, and it can even predict mortality in certain diseases. Or another example, which is interesting and has been ignored for years and years, is that we have lots of fluctuations in the normal capillary hemoglobin oxygen, so oxygen saturation of blood. We can measure it non-invasively because SpO2. And thanks to COVID, of course, we all know how to use it. The general public know how to use it. So these normal fluctuation that has been around and we can see in health and disease hasn't been studied that much. So in the past few years, basically, so some of my very good friends and colleagues and previous students from our lab, including Amar and my collaborators at the University of Portsmouth, which I have the honor of collaborating with them, basically, they have done lots of interesting studies. For instance, if we look at these fluctuations, why we have fluctuation, does it give us information about the integrity of cardiorespiratory system? Probably yes. How can we measure these variations? We can use entropy analysis, for instance. And so Amar Bogal, Dr. Bogal, who is now a clinician and a previous graduate from our lab, basically, so he showed that if you measure entropy of this fluctuation, it gives us lots of information about the cardiorespiratory system. Why he used entropy? I want to just summarize his reasoning, why he used entropy analysis. Why? Because entropy analysis of time series is a very good measure of measure of information content of a signal. Basically, if you think about the foundation of information theory by Claude Shannon, which revolutionized how we live and all of the technology which is associated with our lives, it is based on quantification of information. So basically, we have exchange of information between physiological components. Can we use entropy to measure it? To understand how information is linked with the content of information, I want to show you this cartoon. Basically, here we have a grandma and grandkids, for instance. The kids are trying to talk to the grandma. And of course, one of them is saying da, 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 da. Of course, we don't know really what is the message. There is some sort of information, but the information is limited. But the other grandkid is talking about, hi, grandma, I'm playing Superman. So there's lots of surprises. It's irregularity, more information. Basically, Shannon defined in 1950s, 60s that the amount of information, a signal, is associated with average amount of surprise. And because if we have more irregular, for instance, this message, even if we don't know English, this message of hi grandma, I'm playing Superman is much, much more irregular than da, 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 da. So it can contain more information. So if we have such measure, for instance, if we can measure entropy as a measure of information, can we use it in routine clinical practice with signals that are available for all clinicians? Of course, we want to make sure that the methodology is accessible to all people as well. So here is another study by Margaret Gergita, one of our very excellent graduates who is now doing a PhD at Imperial College London. Basically, what she did, she extracted data from mimic data set, which is at PhysioNet, as we discussed earlier. So she looked at patients with sepsis, and she measured the entropy of SpO2 or capillary oxygen saturation at the day of the admission at the intensive care unit. These patients are critically ill, as you know. And then she followed them up. So this is called a Kaplan-Meier graph. The x-axis, which is used a lot in clinical medicine. X-axis is the follow-up time, 30 days follow-up. And then y-axis shows survival. At the day of admission, all people were survived. But some people died, unfortunately. Those people who had higher entropy of their SpO2 at the beginning, at the day of admission, they were more likely to survive. That's interesting. And people who had lower SpO2, some of them died, and you can see a very significant difference between these two. So knowing the entropy of a signal can tell us information about mortality, and I think it matters and important. Why we care about mortality in clinical practice, apart from, of course, trying to avoid and trying to help people, if there is a physiological mechanism that can predict survival, if we understand that, probably it will help us to, of course, understand to understand those positive compensatory mechanism, which of course we are looking for. And those data show that actually, this predictive value is independent of the severity of the disease and all of the common markers that are used in clinical practice. So we have this data, but we have to interpret it. Probably we need to connect it with a kind of a physiological map, similar to those that Professor Ivanov showed you earlier. So if we think that entropy is linked with connectivity and information processing, it is a bit hypothetical. Can we actually map the physiological, maybe a component of the cardiorespiratory system? Can we map the component of cardiorespiratory system to see if those respiratory entropy is actually showing something about connectedness and integrity of physiological system or not? This is a good question. To do that, okay, so let's see what kind of signals are available, and we can measure non-invasively from patients. So this is a slide from one of our collaborative studies with University of Portsmouth, Joe Costello and Tom Williams and colleagues. Basically, so in their lab, which is a very good extreme environment lab, they measure basically, of course, those parameters non-invasively: heart rate, respiratory rate, end tidal volume, minute ventilation, etc., etc. At least these three of these signals can be measured non-invasively in patients in routine clinical practice. For instance, in the intensive care unit, respiratory rate, heart rate, and oxygen saturation is measured easily, right? So we have actually those signals available. Is it possible to map a network where in a patient in intensive care unit, for instance, we can map how much connectedness or how much information is transferred from one signal to other signal? Is it possible? If we can do that, does it have any value? Does it help the patient either in terms of prediction of mortality or deterioration? This is something that we have been busy with many of excellent students and graduates to address really. So I think the idea is, imagine if I have respiratory rate, I have a signal respiratory rate, maybe 10 minutes, 20 minutes, or SpO2 time series of 10, 20 minutes, and heart rate 10, 20 minutes. Imagine. I have these time series. If I can find out a way to see how much information is exchanged between respiratory rate and SpO2, or between respiratory rate and heart rate, that would be very, very good. Let's see if it is possible. Of course, there are lots of different methodologies. One of them, of course, if you collaborate with physicists or mathematicians, they give you lots of ideas. So you don't need to only collaborate with clinicians. You need to collaborate with other people who know about other disciplines. So the idea is, imagine if I have a time series of respiratory rate and I have another time series of heart rate, right? And these are parallel from the same patient or from the same person. We know that physiologically, respiratory rate should affect heart rate. This is called respiratory sinus arrhythmia. It's a very well-known phenomenon in physiology. So basically, if I want to quantify it, there is a way. One way would be, if I can see how past of, at a given time, how past of this time series affect future of this time series. If I can quantify that, it is actually useful. So it can tell us how much information is exchanged, or how much information from X time series, in this case, for instance, respiratory rate, will affect the future of our heart rate. So it is a way to measure transfer of information. And actually, of course, around 24, 23 years ago, a very interesting indicator has been developed, which is called transfer entropy, that can do that. And so if I measure transfer entropy from X to Y, it tells me how much information I've got is transferred between these different signals. Now, if I go back to my previous slide, basically we have respiratory rate, SpO2, heart rate. If I apply this and I try to see how much information is exchanged, definitely it is a directed transfer of information, because the information from respiratory rate to heart rate is different from the information from heart rate to respiratory rate. So I can plot a directed network, which can help me to actually understand the network better, and hopefully to map the network. This is something that my very good, of course, collaborators and very good fellows in our lab, such as Cecilia, who's going to give a talk later today, and not apologize today, tomorrow, is of course has done. So basically, they try to map these maps in patients from who were admitted in the critical care unit of hospitals. These data comes from mimic data set again. And here we see that, okay, if we compare people who have sepsis and we look at people who survived and people who didn't survive, people who unfortunately passed away in the intensive care unit. If we look at these maps, so these links show the transfer entropy, how much information is exchanged. You see that, interestingly, in patients who didn't survive, we have less amount of information transfer. So the connectivity and engagement of system is less, and you can quantify it. And basically, for this can be potentially be mapped in any ICU monitor as well. So that is interesting. And this prediction of mortality from looking at these maps, it is independent of the indexes that are commonly used by clinicians. Very interesting. What is the application? Can we use it is it really useful? If you are going to predict mortality, probably it would be better to predict deterioration, because then we have time to do something for our patient. And in this case, we look at deterioration. Of course, it is a study by our collaborators, from Bart's Matt, who did a great job. He's one of the consultants of anesthesiology at Bart Hospital, as well as Wati, one of our very good collaborators at UCL and Cecilia. So what they did, they tried to see what happens to these patients, which patient deteriorate and which patient don't. 48-hour deterioration can be predicted, and it is interesting. And those people who have no deterioration, we have much more transfer of information. The integrity and the amount of information transfer between these nodes is higher. So this is actually very useful as well to know. So it might hopefully give us time to help these patients if we can predict that who is going to have, of course, to deteriorate in the intensive care. So I want to just summarize what I have mentioned, and I have a few other slides to discuss about other types of network mapping applied in clinical practice. So far I mentioned that network mapping using time series has potential in clinical settings, and probably a disease such as sepsis is important. And we can think about this type of entropy analysis or network mapping in admission to accident emergency units. Of course, the emergency departments of hospitals. It's very important to know which patient might have a worse outcome, so to make sure they are admitted. Signature of catastrophic illness in the intensive care unit, that's important. I touched it a little bit. Prediction of exacerbation of patient with COPD, of course, patient with pulmonary disease, that's very important. Diagnosis of sleep apnea, of course, we are going to have a talk about sleep apnea next week, and my colleagues, very, very good collaborators, will talk about it. And so there are potential applications, and we have to wait and see how data will help us in future. But there is only one problem. One problem we have got is the high resolution physiological signals are not recorded all the time. So if we are in the intensive care unit, that's fine. Of course, patients are attached to monitors. But what about patients who come to our outpatient clinics? What about patients who are at home? How can we map the network of connectivity? How can we use physiological network mapping when we don't have high resolution time series? But for most patients, we have routine clinical data. So can we actually use it or not? Can we use it for physiological network mapping? Or even if it's not a perfect physiological network mapping, can it give us some idea about the network or not? This is something important to discuss now. So to basically to give you this hope that it is possible, I want to refer back to a very interesting study by Azada and colleagues published around 9 years ago. And this group is based on one of the very good university hospitals in Tokyo. And what they have done is they try to see if we can use a kind of network mapping in patient at critical care. And what they have done is, okay, so we have some serum markers, such as, if we want to assess renal function, kidney function, we usually use creatinine. This is routine in all hospitals. We have creatinine for assessment of the kidney failure in patients. If you want to assess inflammation, of course, we measure C-reactive protein. If you want to see, of course, if someone has heart failure or what is how is the situation of the cardiovascular system, we measure, for instance, BNP, brain natriuretic peptide. For coagulation, we can measure D-dimer, etc. So we have these biomarkers that are routinely used and measured in hospitals. What they did, which is interesting in a way, it's simple, but it's very, very nice. What they did basically, they matched two groups of patients, a group that survived intensive care and the group that didn't survive, right? And they matched based on severity of disease and age, etc. So it's a matched data set. And they look at the correlation between these biomarkers. For instance, okay, so we have a data set, we have a population, we have a data set of creatinine. At the same time, we have sodium or CRP in, of course, a population of patients. They look at the correlation, simple correlation. And they saw that we have much more correlation actually between these, of course, nodes of the network in people who survive intensive care unit. And interestingly, in people who don't survive, we don't see that kind of correlation. This is interesting. Probably very experienced clinician, they see these type of things in the intensive care unit, but we never quantify, we never look at the correlation between these data. This is very simple statistics, but very interesting. Can we use this type of network mapping in clinical practice? I want to give you another example of similar approach. For instance, this is about liver cirrhosis. Liver cirrhosis is a chronic liver failure due to inflammation that leads to liver fibrosis. My very good friends and colleagues, Professor Monteniese and Dr. Alireza is going to talk about it later next week as well. So cirrhosis is interesting because it is a disease with lots of multi-organ involvement. So patient with liver cirrhosis, they have hepatorenal syndrome. It means that the kidney stops working. They have hepatic encephalopathy. They have problem with the nervous system, hepatopulmonary syndrome, cirrhotic cardiomyopathy. They have lots of different, of course, complications. Let's see if we can use the approach that Azada used, and they use routine data to see how the map is different in survivors and non-survivors. So in this case, of course, when we use such kind of mapping, a node of a network is a physiological or biochemical parameter, of course, creatinine, sodium, etc., etc. And the link can be the significant correlation. If there's any significant correlation between the nodes, of course, then we use an edge, and the edge represents association. If there is no edge, it means that there is no significant correlation. And so this is based on data collaboration between our lab, and this is the work that the Tope has done with collaboration with Sara Monteniese in University of Padua. Basically, we see the same pattern that Azada actually described in patients with sepsis and in critical care. Basically, if we look at people who survive and people who don't survive, we have much more correlation between these biomarkers. That's so interesting. So, of course, it means that lack of this correlation is associated with mortality. Interesting, very interesting. It helps us to understand the disease much, much better, and why some people cannot make it, some people cannot really survive such, of course, complex disorder. But there is only one problem. The problem we have got with this type of mapping is that these are population networks. For instance, when I look at these networks, this schematic network, it is based on population. We need to have a population of patients, and then you need to look at the correlation between the data. What if I want to have knowledge about one patient? You clinician look after one patient at a time. So I want to know what is the state of the network of this individual patient, not necessarily all of the population at the same time. Can I use a method to have information about the network of this individual patient without just, of course, having all of those very, very sophisticated time studies? So by just looking at the routine clinical data, such as biomarkers that I mentioned. Fortunately, yes, there is a good way of doing that. And of course, this method originally is by Zanin and colleagues, and later my friends and colleagues, Tope, Oilade, and colleagues, they just make them appropriate and suitable for normal clinical practice. So the idea is, so we want to have individual maps. So if we have this map, let me find my cursor and show you. Okay, so we have this map of population map. The population map means that, okay, so between biomarker A and B, there is a correlation, right? So now if I have individual patients, such as this patient in red and this patient in green, now I can actually measure how far they are deviated from this standard correlation of the reference population, right? And so I can look at it for biomarker A and B. I can measure this distance or this deviation, right? I can do it for other angles, other axes as well. For instance, for the correlation between biomarker B and C, we have a correlation. Again, for patient who is red and the patient who is green, we have basically a deviation from it, and we can measure this deviation. If for each individual patient, I measure how much they are deviated from this axis, basically I can plot a network, and this network tells us that how much individual patient is deviated from their reference population. And in this case, actually, we are going to have a network map for individual person or a patient without actually, of course, needing so much difficult data to obtain, because data such as routine clinical data is easy to obtain using normal clinical practice. So this kind of network is called parenchylitic networks. So in Greek, parenchylitic means deviation, basically. So basically, you are trying to measure the network based on how much deviated they are from a reference population. So I almost finished my talk. Apologies that it took a bit longer than usual. I just want to show a few slides to conclude. So if we use this type of parenchylitic network mapping, we can basically, of course, predict mortality very well. It means that we will have data from individuals for prediction of mortality. And in this case, in this patient with cirrhosis, for instance, we can see that we can, based on how the network look like, we can predict who is going to, of course, have better survival than the other group. And the summary of the talk is that, yes, clinical network mapping using routine clinical data is possible. We can either use functional networks, such as networks that, of course, Professor Ivanov showed us at the beginning. So sometimes by using high-resolution time series, we can map different networks, and hopefully they are useful in clinical practice in future. Or if we don't have that much time series, and if we have a reference population, we can actually use parenchylitic networks. It means that we look at how our individual data from a patient or from a person is deviated from a reference population. So it is also possible, and the methodology is being refined, and hopefully it will be used in future as well. So the last summary is basically what I want to sum up what I mentioned in the past 20 minutes. So network physiology has potential in providing information about pathophysiology of disease. But this area is multidisciplinary. So we need to help from many, many different disciplines. So, okay, and I just want to mention before we, of course, we go to for question and answer session, that basically, of course, this session was the first session of the series. We will have 3 more sessions. Tomorrow, we will have a session about network physiology of extreme environment. I hope you all, of course, come and enjoy the session. Very interesting session. Then next week, we will have a session on network physiology of respiratory disease and critical care by Dr. Amar Shah and Emil Ito, very, very good collaborators and excellent scientists. And on 3rd of April, we will have another session on network physiology of liver failure. So, and of course, I want to thank so many people. So none of these work can be done by one person. We have a big team from many different universities who have helped us doing the work. So thank you for your attention.
Thank you so much, Dr. Mani, for expatiating so much about the application of network physiology in medicine. I think because we don't have a lot of time, so I'm just quickly going to jump into some of the questions that we've got. I think fortunately we have just 3 questions. And obviously, the first 2 questions are not kind of specific, but the rest of the questions are quite specific to Professor Ivanov. So the first question is by Hassan. Hassan asked, are there any databases with different physiological data that can be used for retrospective analysis? I think that question is to the floor. I don't know who wants to take the question, the answer. Thank you.
There are large databases in the area of sleep medicine, very, very large, by the way, and also various sleep disorders, certain neurological disorders, and now growing in the context of cognition, because people start to be aware and collect not only cortical data during cognition, but also other physiological signals and parameters, especially studies related to stress, also to memory. And exercise physiology now is growing, and actually there is a company, even Biopac, which in the context of ambulatory and laboratory conditions, you can devise various type of protocols to study patients, let's say under different type of exercise, measuring multiple channels of muscle activity, respiration, cardiac, and so on. It's still, there is a limitation, of course. This is one of the 3 pillars of network physiology is, of course, the devices that can record in a synchronous way, high frequency data, and the second is, of course, the availability of the data. So this is something that the society in general, the researchers and clinicians, we have to start paying more attention, because the more data is available, more discoveries will be made. The same way like it was done with the human genome.
Thank you very much, Professor Ivanov, for that very, very good answer. I'll go to the next question. I think this is absolutely directed to you. Williams asked specifically to Professor Ivanov that thank you very much for your interesting presentation. It would be nice to see how these network activities vary amongst inhabitants of endangered regions versus non-endangered regions. The knowledge will be helpful in making informed decision on health aids sent to victims of endangered regions. And let's say, what do you think about that specifically to you, Professor Ivanov?
Well, I'm not sure what endangered region is. Is this a region where people have war, or what is endangered region? I assume that conditions of stress and would definitely affect the interaction between systems, both dynamically and metabolically. And we have done certain preliminary studies where we study cognitive function at rest, and also just rest without cognitive task. And the cognitive task imposes certain stress. And this is reflected both in muscle response, cardiac, respiratory response. So I would assume, but it's a good question.
Thank you very much again for that. The next question is also directed to you, Professor Ivanov.
Can I just interject something?
Yes.
I think we might even think about, something like climate change and extreme environment, right? So maybe can we apply network physiology to see how we are when we are exposed to extreme environment, because climate change can expose some of us to extreme environment. So for that, I want to invite our friends to join our next talk, which is tomorrow, which is exactly about extreme environment. Apologies for interruption.
No, absolutely. Thank you very much, Dr. Mani, for that. So please join us for the extreme environment tomorrow. Lots of it will be discussed. I'll move to the next question because we don't have a lot of time. So this question is also directed to Professor Ivanov. Thank you for this absolutely fascinating. So that's Luca is asking these questions. Thank you for this absolutely fascinating presentation and such impressive work. I have questions regarding the calculation method of time delay stability. Specifically for Professor Ivanov. This topic is new to me, so I hope my question is not out of scope. When studying the relationship between two signals, two organs, the TDS, that's the time delay stability calculation, relies on identifying the position of the maximum correlation between these signals to quantify the delay. Based on your experience, is this maximum correlation usually clearly defined, or can it be more ambiguous? For instance, because of the presence of several local maxima of similar amplitude. Thank you very much for your answer.
Yes, so very, very valid. This is obviously somebody who has a certain experience with signal analysis. So we went to this approach with time delay stability, looking at the location of the maximum of the cross correlation for a very good reason, because it turned out that cross correlations do not work for physiological systems. So I don't know whether it's well known, especially in this community, but if you have two systems which generate output signals which are correlated, and if these systems are not coupled, but generate output signals which are autocorrelated, if you cross-correlate them, you will find a spurious coupling, spurious cross-correlation. So therefore, comparing also to other entropy measures and other things, this time delay stability turned out to be useful. We have a challenge here that we have to see crosstalk between diverse systems that work on different scales and have different degree of noise and so on. So therefore, yes, in most of the cases, you may have a problem with identifying if you have several local maxima, but this may be an individual, usually an individual bursts in a signal. If this would not, you will not encounter the same situation multiple times. And if there is a real coupling between systems from point of view of a fixed time delay, then in the majority of the cases, you have well-pronounced burst, and then you see a relatively well-pronounced peak.
Absolutely. Thank you very much. Thank you very much for that.
It's very
For that,
good.
yeah, absolutely. So, for the final question before I wrap up, Julia, oh no, there's more questions coming in, but we'll try to say. So, is there any example of application of network physiology in cancer?
Oh, yes, people start, I'm aware of papers. If you look actually at this journal, Frontiers in Network Physiology, they have been recently in the last 2-3 years, couple of publications, including group from Milano, University of Milano also. Right now, there are technologies, imaging technologies being developed to identify cancer simultaneously, multiple systems across scales. So both on the computational and on the imaging side, network physiology has made inroads in cancer research.
Thank you very much. And thank you.
Can I apologize? Can I just
yes.
mention?
Yes.
So I think to the application of pancreatic networks, there are very good papers on ovarian cancer, etc. So, and then if our friends who ask this question, if they type for pancreatic networks and ovarian cancer, there are very, very interesting papers about this kind of network mapping in cancer. But also there are very, very interesting studies on if we want to predict, if someone has, of course, cancer and they undergo therapy, for instance, chemotherapy or bone marrow transplantation, if they are susceptible to develop sepsis. So how we can predict sepsis in those patients to prevent any catastrophe. Of course, there are studies on that as well.
Thank you very much, both, for that very good answer. I think Lucas said thank you very much that you've clarified the question very much, Professor Ivanov. And thank you everyone who contributed on the chat as well, including Danilo, who had put the link to the PhysioNet on there as well for those who want to go and have a look in future. But thank you, everyone. I don't think we're going to have a lot of time to wrap up, but I will take this opportunity to invite most of you and all of you to our tomorrow's session, which is on extreme environment. It's going to be packed full with exciting new talks. And thank you, everyone, again for attending today. And we wish you all the best and good luck in your endeavor. Yes, thank you.
And thank you very much. I also want to thank Abby, Simon, Sarah, and Kathleen from Physiological Society. They are behind all of those technology that we are using. Thank you very much for organizing it.
Thank you, guys. And thank you for all the speakers. Thank you, Dr. Mani. And thank you very much, Professor Ivanov. Thank you.
For the invitation.
Thank you. Okay.